first topic we'll look at is body symmetry. Body symmetry is defined as the balanced arrangement of body parts around a central point, or the balanced distribution of duplicate body parts within the body of an organism. There are three types of symmetry: bilateral symmetry, radial symmetry, and asymmetry. Types of symmetry, so we consider them one after the other. Bilateral symmetry refers to the division of the body along only one plane to produce two mirror halves. Example, in the human body. Radial symmetry refers to the division of the body along any plane to produce two mirror halves. Example, an orange. Asymmetry refers to where there is no symmetry. Example, in the sponges, in the phylum Porifera. Next, we look at sectioning. Sectioning refers to cutting a biological specimen. There are many ways of making sections: longitudinal section or LS, transverse section or TS, and vertical section or VS. We consider them one after the other. Longitudinal section refers to a section that runs through the length of the whole or part of an organism. Example, the LS of boiled egg. Transverse section. This refers to a section that runs crosswise through the whole or part of an organism. Example, the transverse section of the stem of the water leaf. Vertical section refers to a section that runs upright through the whole or part of an organism, usually for an organism with no distinct length or breadth. Example, the human eye. You cannot say it has a length or a breadth. In that case, you refer to it as a vertical section, as you can see. Orientation. This refers to how the body is positioned, or the angle, direction, or position of the organism in relation to the viewer. There are many types, which includes the anterior view and posterior view, dorsal view, and ventral view. We also have lateral view. We consider them one after the other. Anterior view is a view from the front or head end of the specimen. Posterior view is a view from the back or the tail end of the specimen. The dorsal view refers to the view from the top or upper part of the specimen. The ventral view is that view from the bottom or the underside or belly of the specimen. The lateral view is simply the view from the side of the specimen. Now, a microscope is an instrument or apparatus that magnifies a microscopic object or specimen many times to increase the size of the image for observation. So, a microscopic object which is too small to be seen is enlarged. There are many types of microscope, but this course considers only the light microscope. A light microscope is the type of microscope that works on a principle that light rays passing through a small transparent specimen is refracted by lenses to give an enlarged or magnified image. That image is further magnified by two more lenses. An earlier and simplest form of the microscope was the hand lens. The hand lens is simply a biconvex lens mounted in a frame. It is placed a short distance from the specimen when in use, and the user varies the distance until an enlarged sharp image is obtained. Note, if a lens is used in order to make a drawing of a specimen, the magnification of the lens bears no relation to the size of the drawing. This is the diagram of the hand lens. It has a frame in which the biconvex lens is fixed and a handle. We have generally three types of light microscopes: the simple, compound, and stereoscopic. There you have a diagram showing the simple and the compound light microscopes. A simple light microscope has only a single lens or eyepiece lens and therefore has relatively low magnification. That is a compound microscope. We shall come to the parts shortly. A compound microscope is also called the bright field microscope. It has higher magnification because it uses at least two sets of lenses, an eyepiece lens and an objective lens. It also produces a two-dimensional view of the specimen. Then we have the stereoscopic light microscope. This is also called a dissecting microscope and used for observation with low magnification. The image formed, please note, is almost the same as the one obtained in using a single monocular eyepiece. The principle is that light is this time reflected from the surface of an object rather than being passed or transmitted through it, and it produces a three-dimensional view of the specimen. Part of a compound light microscope. It has a mirror which reflects light rays from the light source through the condenser, the stage, and the specimen into the objective lens. It has a diaphragm also called a contrast, which regulates the amount of light that passes through the condenser to the specimen. The stage holds the glass slide in place and it is clipped. The condenser focuses a beam of light onto the specimen. Both the glass slide and the cover slip are used to mount the specimen. The cover slip is placed over the specimen to avoid wetting the lens. The lenses are of two types. The objective lens, which is held in a nose piece. It magnifies images from low to high power. The eyepiece lens magnifies the image produced by the objective lens further. The focusing knobs are either coarse adjustment or fine adjustment. The coarse adjustment is used for initial focusing of the low power lens onto the specimen. The fine adjustment is used to obtain a sharp image of the object. The hinge allows the whole microscope to be tilted for easy viewing. The limb is used to transport or carry the instrument from one place to another. The base is the part that makes contact with the working bench or table. Handling and caring for microscopes. 1. To carry a microscope, hold the support arm with one arm and place the other arm below the base. Do not pick a microscope by the stage. This can cause misalignment. Lower objective lens down as far as it will go. Do not allow the lens to touch the slide. Ensure the objective lens is cleaned immediately after use if immersion oil is used as mounting liquid. Objective lens, eyepieces, and condensers may be removed for cleaning, but use only lens paper or lens cleaner to clean a microscope. Do not use solvents to clean microscopes and use an aspirator to remove dust. Cover the microscope with a dust cover when it is not in use, but allow it to cool first after use. When you turn a microscope on or off, use the switch, not the power point. And do not switch the microscope on while using full light intensity. After use, rotate the nose piece so that it is always on low power objective. Store your microscope in a clean, dry place away from corrosive chemical fumes. Salt, air, and damp can cause damage to a microscope over time. perform an annual maintenance check by cleaning and lubricating all moving parts. Thank you.